Yes. Yoon-Jun, yes.
Byung-Jun, Dr. Yoon, is associate professor in Texas A&M, Department of Electric and Computer Science. Also, his research has been doing a lot in graphics, Informatics network. So it's a perfect match to our center. And Dr. Yoon also received Ph.D. in electric engineering from California Institute of Technology, 2007. Associate professor, so doing extremely well in Korea. So because I'm late, I'm probably need more time to you, so you can give us a exciting talk. Otherwise, probably I can talk about you. But one thing I would like to mention, Dr. Yoon also received a very competitive NSF Career Development Award, which is essentially in that field, once you receive it, you automatically promote to the next level. Congratulations. Yeah,
thank you. Okay, thank you so much for the kind introduction. It is my great pleasure to be here and then have the opportunity to talk about my research projects. As you can see from the title, I'll be mainly talking about my research projects that involve system-oriented approaches for doing integrative omics data analysis. So you'll hear about system-oriented approaches, network-based approaches, and network analysis a lot throughout the talk. So this is the outline of my talk. I'm going to start with a brief overview of my research projects and then move into three specific projects, where the first one is on integrative omics data analysis with a focus on disease classification, especially cancer classification. The second topic that I will be talking about involves comparative analysis of biological networks. And the final topic is about my latest research, which involves quantifying and handling uncertainty in biomedical setting. I'll briefly mention about some future research plans and then conclude my talk. So let me start with an overview of my research. Basically, I work a lot on probabilistic models, especially probabilistic graphical or network models, and network-based methods that can be used for analyzing biological systems and biological data, especially omics data. More specifically, I've been working on integrative omics data analysis based on graphical models. And then another important topic for me is comparative analysis of graphs or data with topological structure, biological networks, PPI networks, gene regulatory networks, and things like that. Quantifying and handling uncertainty is one of my latest research topics. So I'll probably just briefly touch upon this because I'm pretty excited about this. And finally, my original Ph.D. was on biological sequence analysis, especially on gene prediction, pattern modeling, modeling of RNA secondary structure, prediction of non-coding RNA genes. But I'm not going to talk about this in this talk. So we'll focus on the three topics, which are all related to system-oriented approaches for analyzing systems and data. So there is a coherent research team that are common to all these three projects, and probably, well, the easiest thing that you can see is, well, all of them involve an analysis of omics data, right? And not just omics data, usually they are very big, so big omics data. However, what I want to talk about is not that I'm dealing with a lot of data and big data, but then I had a specific question in mind, which is this. We know we are excited about the opportunity to have and generate a lot of data, big data. However, are they really big enough for the problems that we are dealing with? Well, if you think about the complexities of the living systems that we deal with and the complicated health-related problems that we are facing every day, probably, well, we cannot easily say that the data is big enough for our problems. Well, it's true that they are big. We have big data. However, we are often dealing with bigger systems with bigger complexities. So one question is, how can we, well, use big data to deal with bigger or more complex systems? We need to, when we are looking into complex systems that have a lot of independent variables where many of them are interacting with each other, it's very important to look at their interactions, not just as at the individual molecules or individual variables. So that's the main coherent theme. So since I have mentioned about the coherent theme throughout these three projects, let me start with a presentation of my first research project on omics data analysis. So let us consider the problem of identifying biomarkers, especially gene markers, based on gene expression data. Well, this is a very widely studied problem, very well-studied problem. However, everyone who is working on such a problem also agrees that it is practically still very challenging. One of the main reasons why it's still very challenging is because we are dealing with big data. However, the sample size is still quite small. It is quite small, especially when you think about the potential gene markers that we have to deal with. So we have a large feature set, potentially 10,000 to 20,000 potential gene markers, and we have a sample size of maybe 50. If you're lucky, you may get 200 or 300, right? And then you have to pick the few genes that really make sense to you, and then can be very useful for making predictions. Of course, there is noise in high throughput measurement data, and heterogeneity also makes the problem even more difficult. One very interesting example that I want to talk about is the breast cancer study, and this data set has been analyzed and reanalyzed by a lot of people. So the two data sets, typically referred to as the USA data set and the Netherlands data set, they have profiled the gene expression from about 300 breast cancer patients, and they wanted to look at the gene expression data to try to build up a model of classification model, or simply a classifier, that could be used for predicting their prognosis. Will it be good or bad? Will the patients develop metastasis within 5 years or not? Okay, that was the goal. Because if we can make the prediction very accurately, probably it could be used in making decisions about how aggressive we have to be in terms of therapy, right? Interesting thing is, in both studies, they have identified about 70 gene markers. But the curious thing is, if you compare these two sets of 70 gene markers, they had only 3 genes in common. Of course, theoretically, it's completely possible that both sets, even though they overlap only minimally, can equally well predict the disease outcome. However, we have to think about, what might be the underlying reason if they are as good as each other, right? What's more curious and interesting is, if we use the gene markers identified based on one data set, applied it to the other one, then the classification accuracy was pretty low, probably something slightly higher than 50%, okay? Slightly better than a coin flip. So something's wrong, right? It doesn't mean that identifying gene markers using gene expression data for making predictions is useless. I'm not saying that. It's useful, but then we have to be careful how we use them, and we have to understand the difficulties, especially the practical challenges. So several studies have questioned the reliability of many gene markers that we have been computationally or statistically predicting, and also the disease classifiers that have been developed based on the gene markers that were predicted. And so we are dealing with a difficult problem, and robustness and reproducibility is a big issue. Is there a way to improve them? And the proposed alternative is, why don't we go up one more level? Instead of looking at the individual genes, why don't we look at the system as a whole? At least, if we cannot go to the highest level, why don't we at least look at the genes that are known to interact with each other and look at modules of genes instead of individual genes? So that's what I mean by a system-oriented approach. Instead of looking at individual independent variables, okay, which may not be independent, what we want to do is we want to take this dependency into account when doing the analysis and making predictions. So let me first talk about the pathway-based approach. We want to use the existing pathway knowledge. We know which genes are interacting with which genes, and then they do something together in specific biological mechanisms, right? So why don't we use that to analyze gene expression data at the level of functional modules instead of just individual genes? So we want to do something like this. We look at the gene expression data, and then instead of picking out gene markers, what we want to do is we want to look at the genes that belong to the same pathway, and then try to predict the activation level of the pathway and look at that, use that as the potential marker for making predictions. So it is called pathway marker. So what is one challenge? Well, for individual genes, we know their expression levels, right? Because it is directly measured. However, if you want to deal with a pathway, what you have to do is you have to look at the gene expression level of these member genes and then predict the pathway activity, right? It's an abstract concept. So you need means for doing something like that. And probabilistic graphical models can be very useful for describing the relationship between the genes and the pathway activity level, the pathway activity level with the phenotype of interest, okay? So we know that the phenotype will affect the pathway activities, and the pathway activity of a given pathway will affect the gene expression levels of the genes that belong to this given pathway. However, what kind of data is given to us? We know what kind of pathways have been studied before, right? And we also know the member genes of that pathway, and we know their gene expression levels. So we look at this data, the gene expression data of the genes that belong to the same pathway, use a graphical model to predict the pathway activity, use that for making predictions about the phenotype that we are interested in. So that's the idea. Probabilistic graphical models like Markov networks, Bayesian networks, neural networks, these can be very useful for this purpose. So we were able to show that using a very simple Bayesian network model, we could enhance the classification accuracy. But then what we were more interested in was not the classification accuracy itself, but the robustness of the disease biomarkers, especially in this case, the pathway markers we are predicting based on our method. How robust and how reproducible are they? And suppose we construct a classifier based on the identified markers, right? So how reproducible are these the classification performance? So that was our main interest. So let me show you some results. Well, this shows the discriminative power of the pathway markers that we have predicted. As you can see, this top curve that corresponds to the markers that we have identified. So pretty good compared to all the rest. However, what's much more interesting than these two is the following, okay, these two. So for example, in this figure, we try to identify the top pathway markers based on the Netherlands data set. And then what we did was, we evaluated the discriminative power of these predicted pathway markers on the second independent data set, okay? So what's important is that the predicted markers predicted based on one data set should retain a lot of discriminative power on independent data sets if they are to be used for making robust predictions, right? You can see that, well, this curve is slightly lower than the original one, where markers were predicted based on Netherlands data set, evaluated on the same data set, okay? If we made predictions based on Netherlands data set and then applied it to the USA data set, well, still quite discriminative, right? However, if you look at this orange curve, which corresponds to using just gene markers, the top genes which were predicted to be most useful for making that prediction, how much discriminative power do they actually retain? Well, you can see this flat curve, right? Meaning it can be very, very sensitive on the data set that is being used, okay? So this clearly shows one advantage of taking a more systems-oriented approach. This is the classification performance. You can see that even though our method improves upon the other methods, okay? However, it's not that great yet. It's just about 70%, right? Even if we test it on the same data set. If we design the classifier based on one data set and apply it to a different one, an independent data set, of course the performance was even lower, slightly, around 65%, not too great yet. There's a large room for improvement, long way to go. However, the trend is there, right? One very interesting thing is you should look at these orange bars that correspond to using gene markers, okay? What happens when we rely on individual gene markers without considering their potential interactions? You could get pretty good results if you test your classification algorithm or classifier on the same data set. That's what we usually do, using cross-validation, leave one out experiments and things like that, right? However, if you design the classifier using only single gene markers on one data set and apply to the other, well, you can see that the cross data set experiment performance can be pretty low. So there's a big issue in terms of reproducibility. There's one limitation of pathway-based method. What might that be? The thing is, even though a lot of pathways are known, they don't cover all the genes that we have been measuring, okay? For example, microarray data could have the expression levels of tens of thousands of genes. How many of them are actually covered by experimentally validated pathways? Well, let's say we want to be a bit more flexible, so we allow computationally predicted pathways as well. Still, well, at the time we did this research, only about 40% of the genes were covered by known pathways, which means we are limited by the pathways that we know, which means if we rely on this for our prediction, we may leave out very important key genes, which play really important roles in certain diseases, right? So how can we boost this coverage of genes, okay? So that's where this network-based method comes. So originally, this idea was proposed by a UCSD group by Trey Idekar. The idea is the following. So we have gene expression data. Why don't we combine it, overlay it with protein-protein interaction network data? How do we do that? By mapping the gene or gene expression level to its protein product on the network. So what is the goal? What is the goal? We want to analyze the gene expression levels of the genes that are clustered together, the genes that show coordinated behavior on the network and are very discriminative in the sense that they can be used as useful biomarkers. Well, we want to find such markers, which is typically called the sub-network marker, okay? So basically, conceptually, it's quite similar to the pathway marker. The only difference is, instead of relying on known pathways, we want to actually identify or predict such potential pathways by analyzing the network. So usually, the protein-protein interaction networks that we have cover significantly larger number of genes, okay? So tens of thousands up to 15,000. And if we also include like computational predictions of protein-protein interactions, well, almost every gene could be covered, okay? So we are solving the limited coverage problem. Now then the question is, we know that those sub-network markers could be quite useful for finding robust markers, making more reproducible predictions. But there are two important things to consider. So suppose you have a potential marker. You should be able to evaluate how good that potential marker is, right? So that's one thing. The second one is, well, suppose you know how to evaluate the potential marker. Then how do you find the marker that actually maximizes the discriminative power, right? What is most relevant to the phenotype of interest, right? Classifying the phenotypes of interest. So the original solution proposed by the UCSD group was the following. They used the mean gene expression level of the genes in the sub-network, and then they used mutual information between the mean gene expression level and the class label, for example, good prognosis or bad prognosis. They wanted to maximize this mutual information. And then they used the greedy seed and extend approach to find the sub-network that maximizes this mutual information. That was the approach. In our lab, we proposed a different approach, but with a similar concept. So instead of relying on mutual information, we are using a Bayesian network model, okay? We are using a Bayesian network model to evaluate the effectiveness of a potential marker. We use dynamic programming to find linear markers, okay, linear path that includes a lot of genes that maximize this discriminative power. And then overlapping paths have been merged to obtain sub-network markers, okay? So the resulting markers look like this. These are some of the examples of the markers that we predicted. As you can see, it shows the potential gene interaction, right, in that sub-network. It also shows which genes are, highly expressed in this phenotype or that phenotype. And yes, and these are some of the examples. If we look at the discriminative power of the sub-network markers, it clearly beats the pathway method. And one of the reasons is the limited coverage problem has been, effectively addressed. Okay, so it includes more genes, and it may actually use genes that may be more critical in making the prediction because we are not bounded by known pathway knowledge. Classification performance is also boosted. You can see that it's especially good on the same data set experiment. However, they also have a larger risk of overfitting, okay? Because we have more degree of freedom. So there is a trade-off we have to be very careful about. However, still they lead to more reproducible results compared to other pathway methods or methods that simply rely on single genes, okay? So far, so good. Any questions? Okay.
Is this based on some sort of cross-validation?
Yes, it's based on 5-fold cross-validation experiment.
So each of these bars, you actually have error bars to
That's right, that's right. So I'm not showing it here, but then I'm just showing the average AUC. But it would be certainly useful to show the error bars too, yes. So until now, I have talked about integrative network-based analysis where I use gene expression data with protein-protein interaction network data, right? However, we could use a similar network-based approach even if we have only RNA sequencing data or gene expression data. Okay, so to show how that is possible, let me briefly talk about one of my recent research, collaborative projects with microbiologists. So the goal was this. We are dealing with a fungi called Fusarium verticilioides, and what we are interested in is we want to find gene modules that may be crucial for the pathogenicity of this fungi, okay? So the Fusarium verticilioides is known to trigger disease in various crops, especially maize, so that's why we are interested in this in Texas. And they lead to the crop disease such as the ear rot and stalk rot, as you can see. They don't look beautiful, but then it happens a lot, okay? And another very important problem is they produce toxic secondary metabolites called mycotoxins, especially fumonisin. So it's a big food safety or food security problem, okay? So what did we do? We first sampled maize, which was inoculated with 2 different types of Fusarium verticilioides. One was the wild type. The other one was a gene knockout mutant, which loses most of its virulence, okay? So loss of virulence mutant. So as you can see that the wild type causes a lot of trouble to maize. However, the gene knockout mutant is, well, not that pathogenic, okay? They have significantly different pathogenic potential. So what we want to do is we wanted to compare these two and understand what's happening inside, okay? And that was the idea. So what did we do? We got samples at 3 different time points after 3 days, 6 days, and 9 days after inoculation. And based on these samples, we did RNA sequencing, got the RNA sequence data. And then based on the RNA sequence data, what we did is we predicted co-expression networks to see which genes are highly correlated, okay? So these are the networks that we got. And then we went on to find sub-networks in that co-expression network, which includes genes that are highly correlated to each other, okay? And at the same time, collectively can be used for making prediction about, well, does it come from this highly pathogenic type or the non-pathogenic type, okay? So that we can clearly see the differences between these two types. Bayesian network model was used to evaluate the differential activity of these sub-networks, and then we use seed and extend approach to predict the modules. So these are some of the examples that were predicted. One cool thing about agricultural research is, well, you don't have to worry too much about, whether you'll kill the patient or not, right? If you kill the maize, probably it's okay. If you kill the fungi, well, maybe it's okay, right? So doing experiments is very easy. So these predicted sub-networks are currently being validated through gene knockout experiments by my collaborators. So we've got some initial results this year, okay? They are not complete, but then at least it seems the preliminary results look very promising, okay? So to summarize, network-based, we have talked about a couple of network-based methods for omics data analysis. So network models can play very crucial roles in understanding and analyzing the interaction between the main players, like the genes, okay, in this case. And they provide a very nice basis for integrating data, especially in this case, a protein interaction network with gene expression data, okay? And network-based methods can be used to identify novel potential network modules or potential pathways, so which could be useful for studying human diseases as well. These are some of the publications related to this topic, and my talk presentation was mainly based on these two papers. Okay? So before moving on to our next topic, do you have any questions? Okay, questions?
Yeah, I think another advantage of your approach is that if you do things in terms of pathways, that's something that people can think about and people understand it is the way the biologists think.
Exactly.
You don't think about individual genes, but you think about how they work in the context of pathways. So in your work, where you're trying to find new pathways or new members of pathways.
That's right. We actually also wanted to predict the gene regulations within the predicted pathway because that's more interesting. We can better understand the mechanism.
Yeah.
Yes.
So I'm just wondering, have you had a biologist review some of the results you get to see if they're
Yes, for the cancer data set, it was very indirect. It was reviewed by one of my colleagues, okay, who does cancer biology, but he simply the only thing that he could do was, okay, it makes sense to him, but then it doesn't mean that it's like thorough validation, right? We had to rely on gene ontology analysis, for example. It's like indirect validation because there was nothing better that we could do. However, the Fusarium project, okay, we have the luxury to test whether those interactions really exist. If we knock out the key hop gene in that predicted sub-network, will it really disrupt pathogenicity in that Fusarium, which is really interesting. And the preliminary results we are getting is very, quite promising.
So it seems like you need more cancer colleagues to...
Definitely, and that's why, exactly, that's one of the main reasons why, I'm giving this talk here today. Yes, thanks. So if there are no more questions, let me move on with my second topic. My second research involves comparative analysis of biological networks. I don't know how many people are familiar with this topic. There might be some, and there might be some others who have not heard about something like this. However, it's very easy to understand if you think about comparative sequence analysis or comparative genome analysis, okay? Probably all of you have used BLAST, right? Probably many of you may have used Clustal, right? Why do you use those tools? To compare genes, protein sequences, right? Or genome, right? To do genome analysis. Conceptually, it's very similar. The only difference is, well, computationally, dealing with networks and graphs is much more complicated. So can we implement the same concept on graphical data? So that's the main idea. So to better understand complex systems, we want to look at the interactions. Okay, and recent developments, for example, high throughput techniques for measuring PPI, for example, like the E2 hybrid, or text mining techniques, which have been used to dig the known molecular interactions that are already published, right, have been very, very useful. So setting the stage for more systematic genome scale study of molecular interactions. So these days, we have networks like this. This is the East PPI network. Okay, this is the C. elegans PPI network. They certainly look beautiful, okay? I actually love to stare at them. However, can we do something more? How can we actually make sense of these network data, okay? Can we get some novel, meaningful, significant biological insights by analyzing these data? So that's the question. Okay, how can we analyze available biological networks? Well, you already know the answer. I'm going to talk about comparative methods for analyzing those networks by comparing them, identifying which parts of the networks are conserved, okay? There are two different types, at least two different types of techniques in comparative network analysis. The first one is called network curing. Probably based on the name itself, you could imagine what it actually does, right? It can be viewed as the network version of Google, okay? Or we can say that the network version of BLAST. Instead of searching for genes that may look similar to your own gene, now what you're doing is you have a pathway that you've been working on for a long time. You want to see whether similar pathways exist in the networks of different organisms, okay? So it's like network version of BLAST. So in this simplified diagram, we have a very simple linear query pathway, and we want to search a large target network to find out which part of the network matches this query best, right? So even though this looks very simple, the actual problem is much more complicated. Why? Because the query pathway doesn't have to be linear, right? And when you find the best matching query in the target network, there may be a lot of inserted nodes, deleted nodes, inserted and deleted interactions, right? So if you think about the number of potential mappings, it increases exponentially, okay? It makes the problem computationally quite difficult. Anyway, network querying can be used for searching for non-functional pathways in a new network to find out, okay, are there similar pathways, okay? So this enables us to do some kind of knowledge transfer between different species. You can start with a species or model organism that has been really well studied, okay? And then try to apply this technique to analyze the network of less studied species, okay? And then it can lead to working hypotheses that are very valuable. So these are some of the examples that we got based on applying our network querying algorithm. By querying the East map kinase pathway in the East network, C. elegans network, and the fruit fly network. And these are the obtained results, okay? One thing that is very challenging in this type of research is, okay, these results look interesting, but how do you make sense of this? Do you know whether this makes sense at all or not? It's very difficult to validate, okay? So the thing is, in most cases, people rely on gene ontology for making indirect validations. Okay, that's one of the biggest challenges. Okay, experimental validation is not easy. Other types of validation techniques are also not, almost virtually non-existent. So in our case, after developing these methods, what we want to do is, okay, we are making some theoretical assumptions. Okay, then suppose those assumptions are acceptable. Why don't we come up with synthetic models and synthetic queries and networks and see at least whether it'll work perfectly in such cases, okay? And we've been using a lot of synthetic models, and we have published that data too, so that other researchers can use it as well, okay? Because I know that everyone I met and talked about this have difficulty with validation. Anyway, we also use some human pathways in the CAC database, searched the fruit fly PPI network, found some matching pathways, and they overlap actually pretty well with the putative pathways predicted in the CAC database. So it was very good to see that. The second type of technique in comparative network analysis is called network alignment. And again, based on the name, it's not too difficult to figure out what it actually does. We have networks, we want to align them. And what is our goal? We want to find out whether there are sub-network regions or network modules that are conserved across different networks that come from different species or different conditions, right? In this example, we have two networks. They have two conserved, well-conserved sub-networks. So we want to identify these sub-network modules in an unsupervised way. Conceptually, it's analogous to whole genome alignment. So you could align the genomes of multiple species, and then what do? You look for conserved regions, the regions that have some mutations and changes, however, are conserved very, very well across different species. Why are they conserved? There must be a reason. They could be involved in some very crucial function that is crucial to all those organisms, right? So the same theory and philosophy applies to this case as well. So it could be potentially used for finding novel functional modules like novel signaling pathways, novel protein complexes, and things like that. So we know that network querying and network alignment could be useful for analyzing biological network data and pathways, right? Now let's think about how can we actually implement computational algorithms for doing this efficiently. There are two things that we need to consider. First, everyone knows how to compare sequences. If you have a novel protein, you want to compare it to already known proteins. What do? You run BLAST, right? You do sequence alignment, and then, well, you look at the BLAST bit score or other types of scores that you like, right? However, when you have networks, how do you actually compare them? When can you say that the networks are very similar or not too similar? You need quantitative means for evaluating the similarity, right, between networks. So the first thing that we need to think about is we need to come up with a good scoring scheme, okay, for comparing networks. Now, suppose you know how to compare networks. Now, you want to find the sub-networks in different networks that maximize this score, okay? So meaning you want to find the best matching parts of these different networks. However, I already mentioned that it's a complicated problem. I'll go back to that later. Anyway, let's first think about the scoring scheme. Why are we using this comparative network analysis technique? Just because we are interested in individual molecules, but at the same time, we are interested in figuring out how they interact with neighbors and why are they doing that and what's important about those interactions, right? Meaning, when comparing networks, there are two things that need to be combined. First is, okay, suppose we have two small networks or pathways of genes, okay? We care about whether they consist of orthologous genes, okay? First, we care about the node similarity, gene-to-gene similarity. But what we also care about is how do these genes in the respective networks collaborate with each other or interact with each other, right? So the second level of similarity is topological similarity or interaction pattern similarity. We need to somehow combine them in a sensible way. So that's what it is. And if we think about the number of potential mappings, it increases exponentially. If you have heard about the graph isomorphism problem, actually the subgraph isomorphism problem, it is known to be NP-hard. So in plain terms, there is no optimal algorithm that runs in polynomial time to give you the best solution, okay? So we have to somehow use ad hoc methods or heuristics. So what is a good heuristic to use? What we are proposing in our lab is the following. For comparing networks, we want to use a probabilistic approach, a probability model. Why is that? Because we can never be sure what might be the best match, right? In some cases, there might be duplications of certain modules, meaning that one part in that network may match multiple parts in the other network, right? So we want to take all those things into account. For that purpose, we are using a semi-Markov random walk model, which I will introduce very shortly. Suppose we did the comparison, okay? We know which node should be aligned to which node with how much confidence or probability. We can use it with the so-called maximum expected accuracy approach, which has shown to be very robust and very efficient when combined with simple optimization algorithms like the simple seed and extend or greedy approach, okay? It still works very, very well. So let me first talk about the semi-Markov random walk model. We have here two networks, G1 and G2. We want to compare them. We want to figure out something like this. Okay, this U4, okay, if we had to find the best matching node by considering node-to-node similarity, could be like blast bit score, okay, based on sequence similarity. And at the same time, we want to look at the interaction pattern similarity. If we combine these two similarities, what might be the best matching node for this in the other network, okay? That's what we call node correspondence. How do we find this node correspondence? Basically, we use a random walker which has two legs or 2 feet, okay? On each network, it makes an individual move, okay? At each time point, it can choose one of the neighbors and then make a move. And of course, this leg also does the same thing on the other network. At each time step, the walker randomly moves to one of the neighbors in the respective networks. And once they enter a new pair of nodes like this, well, it decides how much time to spend at that given pair of nodes. If they are highly similar, suppose based on sequence similarity or structural similarity, the walker spends extended amount of time. If they are not too similar, they make a quick next step to move on to the next pair of nodes, okay? Suppose we use this random walk model and run this indefinitely for a long time, infinite amount of time, okay? And then we ask the following question. Okay, how much time do we spend, okay, on average at this and this node, or this and this node? And we are claiming that if we compute the long run proportion of time that we spend at a given pair of nodes, it can serve as a very useful measure, probabilistic measure, for finding out which node maps best to which node in the other network by combining node similarity and interaction pattern similarity. So why is that? Let's think about it. We just defined the long-run proportion of time that is spent at a given node pair, U and V. U belongs to one network, V belongs to another network. Okay? We want to find out what about their overall similarity or functional similarity. In order to find out whether this makes sense at all, to use this as the node correspondence score, let's think about when the score will be high, okay? By design, based on the design of the random walker, we know that this long-run proportion of time spent at these two nodes will be higher if their molecular similarity is high, right? Why? Just because we designed the semi-Markov random walk model so that it spends more time before making the next step if the current pair of nodes are highly similar, right? Just based on design. And suppose they have high interaction similarity. In another word, both nodes are surrounded by similar nodes. How does it affect the score? Basically, it increases the chances that the random walker will visit these two nodes at the same time, right? So when they have high interaction similarity, well, again, the score will be increased, right? So it naturally combines node-to-node similarity as well as their interaction or topological similarity. That's the basic idea. So one beauty of this method is you don't actually have to run this random walk indefinitely, okay? Suppose it's a perfect model to use. However, you have to wait for years until it converges to something meaningful, right? Well, who would use it? The beauty is we don't have to actually run the random walk model itself. It's just a semi-Markov model. So based on Markov theory, you can easily compute this correspondence score matrix using this simple equation. So what is this? H is nothing but the similar node-to-node similarity matrix, which is part of your input data, okay? You have already compared proteins to proteins. You know their sequence similarity, which could be potential orthologs. So you already know that, okay? H is given. What about P? That's nothing but the chronicle product between the stationary probabilities of the two random walks, one in graph 1, the other one in graph 2, okay? And this decouples this random walk very nicely, making the computational complexity pretty low, okay? So computation is also very easy. Now, how do we use this? Our favorite approach is to use this probabilistic measure that compares nodes to nodes in different networks based on this maximum expected accuracy alignment, okay? Actually, I used this approach to build multiple sequence alignment algorithms a lot, okay? So we thought that probably the same principle could be applied to network analysis as well, and it actually turned out to be true. It was very nice. So what is this concept? So what is our goal? We want to find the alignment between networks such that the accuracy is maximized, right? If we know the true network, A star is the true network, then we can compare our alignment, true network alignment. We can compare our alignment with a true alignment, see how accurate it is, right? Of course, this beats the purpose of the whole analysis, because if we know the true alignment, we don't even have to predict it, right? So which means we don't know the true alignment, we don't have the ground truth, so we should measure the accuracy on an average sense, right? That's why we are taking the expectation across all possible true alignments. And how do we choose the alignment? We choose the one that maximizes this expected accuracy, okay? So conceptually, very neat. And now, we have just estimated the node correspondence score, which are proportional to node alignment probability in different networks. How do we use it? Once we have this probability, we can estimate the expected accuracy of a given alignment. Basically, we can count the number of expected correctly aligned nodes, okay, in different networks. So how do we choose A? By trying to maximize this measure using any kind of optimization technique. And in our case, we tried very simple ones like the greedy optimization technique. Even with the simple greedy optimization, which is computationally very efficient, it works beautifully well. So this is our algorithm that uses the semi-Markov random walk model coupled with this maximum expected accuracy, the MEA approach. Let me just briefly talk about its performance. Look at the blue bars. As you can see, the querying results are pretty accurate, especially compared to all most of the state-of-the-art algorithms that we compare to. And what's really nice about this is because of the decoupling of the random walk that I have already mentioned, it's computationally very efficient. So based on the current PPI networks that contain about 50 to 60,000 nodes, 5,000 to 6,000 nodes, up to 15 to 20,000 nodes, with hundreds of thousands of interactions, and queries of size between 10 to, let's say, 30, well, the computational time for querying is typically less than a second. However, if you compare that with competitors, the other most popular algorithms at that time, usually tens of seconds up to minutes, okay? So it's extremely efficient, at the same time, the most accurate algorithm. So you can see the power of taking a probabilistic approach coupled with this maximum expected accuracy criteria. We also applied this technique, the semi-Markov random walk model, for comparing networks, and MEA approach for constructing the alignment for solving the network alignment problem. And these are some of the examples, and they overlap, and these have been obtained by aligning the fruit fly, human, and yeast PPI networks. And these are the predicted results, and they overlap very well with known protein complexes. So it's one type of evidence showing that this type of approach could be quite useful. Okay, I'm not showing the computational time and accuracy because the trends are very similar. But let me just mention that it enhances the accuracy a lot, but at the same time boosts the speed at least 100-fold, which is pretty amazing. We are very happy to see the results. Anyway, let me summarize. We want to understand biological systems by looking into the interactions between the variables or the molecules that are involved in that system. And given genome scale networks, well, I've been mainly dealing with protein-protein interaction networks or co-expression networks, but then it could be something completely different. Using comparative network analysis tools like network querying, network alignment, we can make predictions about potential network modules, and then, well, thereby come up with nice working hypothesis about how genes, how proteins interact to do something important that is important across different organisms, okay? And we propose to use the Markov, actually semi-Markov random walk model and the maximum expected accuracy approach for quick and accurate results. These are some of the papers related on this topic. Well, it has been one of my main research topics of interest because, well, networks are so flexible, and there's a lot of different types of big data with topological relationships. For example, I could use the same method for analyzing social networks, and actually I tried to do that. So I was analyzing the internal social network of some big company, okay, trying to find out who might be potential people who breach data, okay, private data. And it was very interesting that we could use very similar tools to other types of networks as well. Anyway, in this presentation, I mainly focused on these two papers, and there is a lot of follow-up work, okay? Any questions?
So, you have a network that is weighted and directed instead of just protocol network.
Yes.
Would that, will your method
Yes.
be used for that? Or?
That's right. That's a very good question. And actually, yes, it takes that into account very nicely because the network model, we can set the Markov transition probability matrix so that it follows only the directions of regulation, okay? So it can take care of directed graphs. And then, what was the second question?
The weighted.
Oh, yes, yes, the weights, if different edges have different weights, we can use that to, to adjust the transition probabilities to the neighbors according to the weight. So it is a pretty general framework, but the only reason why we didn't show that is, well, we don't have such data sets. So it's,
I do.
yeah, oh, you do? Excellent. I would be very interested in looking into that. Okay. But it's, yes, it's possible. Any other questions? I think this part will be the shortest, and it's also my latest research. Less results, but then more excited because I'm doing something very unique, and I hope and I think you will agree that it's very interesting. Okay? So we are interested in robust strategies for handling uncertainty. We know that biological systems are extremely complex, and if someone claims, give me 1 terabyte of gene expression data, I'll tell you completely how the underlying gene regulatory network looks like, okay? Would you believe that guy? Of course not, right? He may get something very useful and effective, right? Very accurate. But would you believe that it'll be the true network? Of course not, right? We are dealing with bigger systems, right? And the big data usually pales in comparison to the complexity of the system that we are dealing with, okay? So measurement data, they could be big, they are useful, however, still provide, in many cases, only limited snapshots about the system that we are dealing with. Which means, well, there is always uncertainty. We have to live with that uncertainty, so which means we have to learn how to live with that uncertainty, how to deal with it, how to quantify it, how to handle it, how to manage it, right? That's the question. And the methods that I've been proposing and reviewing in the first 2 parts of my projects, they can be viewed as attempts to reduce this uncertainty and thereby make better predictions, right? By doing a more system level analysis. Suppose we are interested in inferring the biological network, like the gene regulatory network based on gene expression data, okay? We could start from measurements or gene expression data, and if we have prior knowledge about which genes regulate which genes, what are the pathways that might be part of the gene regulatory network, we could combine that prior knowledge with the data to make the prediction, okay? To predict the true gene regulatory network. So do we actually end up predicting the right network? Of course, we can make our best guess about if we have to find out what's my best guess, this is what I have. Well, I could do something like that. But can I be sure that it's actually the true network? Of course not. So what we get is not the true network, but then a class, or uncertainty class of potential networks which are consistent with the data that we have, and at the same time consistent with the prior knowledge that we have, right? Agreed? So based on data and prior knowledge, what we can actually predict is an uncertainty class that includes all the potential network structures, okay? So we have to live with this uncertainty. Then one important question is, well, you want to figure out how much uncertainty you have. You want to quantify uncertainty. If you are familiar with information theory, suppose you have a bunch of random variables. You want to find out how much information content you have in those random variables. What do? You use entropy. You use information theory, measure the entropy or the uncertainty. If the entropy is large, it means there is a lot of uncertainty. If the entropy is 0, it means that, well, it's not a random variable, it's just a constant, right? Can we use something like that? We could try, and there have been attempts like that too. However, what I'm claiming is probably that's not the right way to handle with uncertainty. Why is that? Practically, why do we care about uncertainty? Because there is a specific task that we want to do. What is it? For example, you may want to use this data, predict the network, to predict your best possible druggable target, right? Or it could be you want to use this data, infer the network, and then use it for finding critical pathways, or using that to build better classifiers, right? Usually, well, in medicine, we have a specific goal. There's a specific disease that we are dealing with, and we want to do something based on data and research, right? So from a practical perspective like that, what we really care is not the uncertainty itself, but then how this uncertainty actually affects the operation that we want to perform. Okay, is that clear enough? There's a goal. We have an objective. So we should be quantifying the uncertainty based on how it affects our mission or goal. That's the whole idea. Okay? Then the next question is, okay, that's great, but then how do you actually measure it? Entropy? Of course not, because entropy has nothing to do with a specific goal or aim, right? It simply measures the uncertainty in terms of how variable your random variable is, right? However, we want to somehow have an uncertainty measure that is clearly connected to a specific aim or objective. Okay? Based on that motivation, we proposed what we call the mean objective cost of uncertainty. It looks like this. Okay? Suppose theta is the true network model, true gene regulatory network, and of course we don't know what it is. But suppose theta is that true network. Okay? Suppose this Xi or Xi for that network tells you, okay, if this is the true network and we want to build a classifier for data generated by that true network, okay, then there is a cost function involved. In this case, if we are interested in building a classifier, we would be interested in minimizing the classification error, right? So this could be the classification error. If this is the true network, what is this? This is our operator, in this case, a classifier. Do we know what the true network is? Of course not. We are dealing with a whole bunch of networks, which might be all true. They belong to this uncertainty class, right? So how do we have to choose our operator or robust classifier? By choosing the one that performs best in an average sense, right? So that's why we call it a robust operator that retains very good performance across all potential networks in the uncertainty class that we have, right? Now, suppose you had perfect knowledge about the system or network, right? What would you do? Well, you don't have to use a robust operator because you have perfect knowledge about the underlying network, right? What do you have to do? You have to build a classifier that is optimal for the data generated by this true network, right? However, can we do that? Of course not, because we don't know the true network. So there will be an increased cost, in this case, increased classification error, just because we don't have perfect knowledge about the system, right? There's this increased cost. Where does it come from? From uncertainty, right? And we have a very specific goal in mind. In this case, let's say prediction, classification. Because we don't know the true network, we have to compute this increase in an average sense. Well, we have to compute the expected increase, right? So there's expected increase of operational cost, which is coming from using a robust operator that has good performance, reasonable performance across all networks that are possible. However, this increase comes from the fact that we don't have perfect knowledge about the true underlying network, right? So it's one way for measuring the uncertainty that is closely tied to the mission that we have, okay? So we call it the objective-based uncertainty quantification method. So MOQ measures the operational cost that is induced by the uncertainty, and it provides a very effective way of quantifying the amount of uncertainty that actually matters to us. Because suppose you're dealing with a very uncertain system with a lot of uncertainty, the variables have high entropy. However, you can exactly pinpoint what is the part of the network that you have to control to get the optimal performance. Then you don't care about the uncertainty at all, right? Because you can perfectly achieve your mission. So that's what it is about. How can it be used? I've talked about methods, network-based methods that use graphical models to combine different types of data, right? But in a sense, it is still ad hoc because suppose 10 years later, someone came up with a really nice experimental device that gives you a type of data that you never imagined before, okay? How can we integrate this with existing data like RNA-seq data, metabolomics data, pathways, networks, right? We have to come up with new heuristics, build new models, right? However, if you think about this method, the only thing that we need to do is, well, if we have different kinds of various types of data and knowledge, we only have to figure out how to use it to define the uncertainty class of networks, and then use the data that you have to define the prior distribution. So it's a Bayesian method, okay? You can use the data and use the knowledge to define the prior distribution of the true networks that might be possible, okay? So it provides a very nice way of integrating knowledge and all kinds of data. Not that I'm saying that defining the prior based on new type of data is trivial, it's not. However, at least we know what is the right thing to do without relying on very fancy, ingenious ad hoc methods, okay? We could use it for designing robust operators, like, well, how to find the most robust drug cocktail that is expected to have the best result on the patient, okay? Or building the best robust classifier, genomic-based classifier for certain diseases. We could use that. We can also try to find out, okay, we have our current knowledge. Which part of that knowledge is really crucial in achieving our mission? What part of the network or system is really crucial? We could find out what are the essential structures or substructures. For example, in the gene regulatory network we have, what gene is most critical in achieving our mission? Which gene regulations are most critical, okay? We could do that. Finally, it could be used for optimal experimental design, and I'm extremely excited about this. This will be my last topic. Why is that? Since many of you, I'm not sure whether all of you, but then many of you will be computational biologists or bioinformaticians or statisticians, you may have this type of experience. You found a new collaborator who has a big data, and your collaborator wants you to do some analysis, right? How does it work? They did what they could based on designing the experiment, did the best experiment, got data, and are asking you to analyze them, right? However, how do you know that there was actually the best experiment that could be done, right? Suppose your collaborator asks you, okay, so we got this initial very exciting results. However, I want to move on. So if I were to design additional experiments and perform them, what should I do? Suppose you were asked that question, what would you do, right? So I was thinking about this, how to find out how we can optimally design experiments to achieve the goal of interest. Well, that's when I came up with this idea. So in order to see how this mean objective cost of uncertainty can be used to design optimal experiments, let us think about the following scenario. Suppose you have a gene regulatory network, and suppose you know which genes regulate which genes, okay? You know about the gene regulations, you know all the edges. However, for certain gene pairs, you don't know whether the relationship is upregulating or downregulating, okay? Just a toy example. So because you have limited resources, you want to test the unknown regulation that is most important to you, okay? Of course, if you have infinite budget, you could test all of them to complete your knowledge, right, about the gene regulatory network, but you cannot do that. So then how can we prioritize the potential biological experiments? How can you make your decision in an optimal way? The idea is this. Suppose this is the ith regulation that is uncertain. You want to find out, it's a binary variable because it's either upregulating or downregulating, okay? Suppose you decided to do an experiment to find out whether the ith regulation is upregulating or downregulating, okay? Now, once you perform that experiment, you will get some result, right? It might be upregulating or downregulating. Then, of course, it could be either one. So you have to consider expectation. Expectation of what? How much uncertainty still remains after performing that potential experiment, okay? Of course, in the worst case, you don't know, you cannot predict the results. You can make them just, equal probability, okay? And then what do? You estimate the remaining uncertainty after potentially performing that experiment, right? If you can compute this measure, it is just straightforward how you should prioritize potential experiments. How do you do them? How do you do that? Choose the one that will reduce the uncertainty maximally, right? That will lead to the minimum remaining uncertainty, okay? That's what you do. So we did some simple experiment based on this mammalian cell cycle network that is pretty well known. And blunt arrows, they show suppressive regulation. And then these regular arrows, they are upregulating relations, okay? We randomly chose K regulations and then assumed we don't know about them, okay? And then what did we do? We wanted to prioritize the K-potential experiments. One important thing to remember is we want to choose the experiment such that if we perform that experiment, it is expected that we can boost the performance of the operator that we are trying to build. In our case, we wanted to minimize, after network intervention, we wanted to minimize the probability of being in a cancerous state or decycling the retinoblastoma and P27 were all off, okay? That was our goal. Suppose this is our objective. What is the experiment that we need to do first? Okay. So we used our framework to make predictions, assuming 5 regulations are unknown, and these are the results. For example, we are comparing the intervention performance between the best predicted experiment and the second best predicted one. How much gain do we have? You can see that we actually have consistently positive gain, and when there is more uncertainty, it increases, okay? If you compare the top experiment with the worst experiment, then this gain will be even larger because we are comparing the best one with the worst one, right? That is predicted. Now, suppose we use this to decide which experiment to perform next, okay? So we were interested in evaluating the success rate, basically the relative proportion of time that the best predicted experiment was indeed better than all the other competing alternatives, okay? So for example, this is the first, the network after performing the best predicted experiment. This is after performing the second best expected experiment. And how do we define success? If performing this lead to better network intervention performance, okay? In most cases, well, we were successful, and the success rate actually increases when there's more uncertainty, okay? Although not monotonously. And failure means that actually the second best predicted experiment was better than the top predicted experiment. It happens, okay? We cannot be always successful, but still we are more successful than we feel, okay? And this was the number of times, okay? So as you can see that this framework could be nicely used for making predictions and making an informed decision about, okay, now I'm at the stage of planning for my future studies. What is the type of data that I need to get if I want to fill the critical gap? What is the critical gap? How do you answer that question? The UQ framework that I just proposed, well, could be used for doing the purpose. So that's about it. These are some of the publications that are related to this UQ work, uncertainty work, and my presentation was mainly based on these 2 papers, okay? Now let me wrap up. So what are my future research plans? First of all, well, I like these beautiful networks. I will continue to work on them. Basically, there is still a long way to go, okay? We could improve it significantly. We are currently working on multi-state random models, which was inspired by multi-state hidden Markov models, which were so useful for gene prediction and genome comparison, okay? So well, we are working on this, and the results look actually quite good. The paper for this work is under review, second review, and I think I'm hoping that it will be published very soon. And of course, this latest research on objective-based uncertainty quantification, I plan to continue to work on this, especially find specific applications related to specific diseases, okay? That'll also depend on what type of collaborators I'll be able to find who are interested in trying to apply this, okay? I didn't talk too much about sequence analysis, but my PhD was on sequence analysis. It involved a lot of RNA work. And currently, this is a project that I just started. I call it the massively parallel bioinformatics. What is the reason I call it that way? If you have used and learned about biological sequence analysis, like sequence alignment, the optimal methods that people use requires the use of dynamic programming, okay? If you want to use HMM, you need to use dynamic programming. You want to use Smith-Waterman? Well, you need to use dynamic programming, right? So BLAST is actually a very nice heuristic method that tries to mimic the behavior of those optimal methods. So for parallelizing, well, dynamic programming isn't that nice. It's actually quite difficult, okay, to parallelize this in general. So what I'm interested in is, can we create message passing algorithms which can be very easily parallelized and which are really suitable for HPC environment by redeveloping all those fundamental bioinformatics algorithms based on a message passing approach. I tried only the sequence alignment for a sequence pair, and it seems that I was able to beat the para-HMM based method, which is the it's not state of the art because it's not new, but that's still optimal, okay? And accuracy was actually slightly better than HMM. The complexity was way, way lower than using dynamic programming. So I plan to work on this too. What I'm also interested is, I only have a very little experience working on public health related problems, okay? I have just one presentation and two conference presentations and one journal paper. However, I'm really interested in applying these network-based methods for analyzing public health data, like EHR, okay? There must be a lot of hidden factors and their relationships, which might be really multivariate. And if modeled and analyzed properly using network-based methods, we could find out very interesting results. That's my expectation. Also using probabilistic graphical models and machine learning techniques for analyzing EHR and health-related data, especially for predictive modeling of health events. That's one of my research projects that I'm planning in the future as well. Let me thank my collaborators and my lab members who contributed to the work that I have just presented, and also the funding agency who supported my research. And of course, thanks to you for your attention. It was a long talk, I know, but I had to talk about it because these are projects that I'm really excited about. Thank you so much.
Okay, since it's quite behind the schedule, probably will ask one more question if you have. Anyone would like to ask one question? Yeah.
So for your last part,
Uh-huh.
how do you update? Like, for example,
Uh-huh.
you use the existing information to predict the
Uh-huh.
experiment to do.
Uh-huh.
Then there are many papers published every day.
That's
and
right.
That will impact your decision.
Exactly.
How do you typically update?
So I didn't think about, it's a very good question, very important practical question, but I haven't thought about the online or on-the-fly update strategy. But it could be, what I'm thinking is, for defining the uncertainty class, prior knowledge, especially in terms of previous publications, will be very important. So probably linking this idea to use text mining tools to automatically update the prior, that might be very useful. But I haven't done much work with respect to that.
Okay, yeah, thank you. Yeah.
Okay, thank you so much.
Yeah.
Thanks.
Yeah, very nice.